Phase 1 trials are the first studies of a new medicine in humans. The primary aims are safety, tolerability, and pharmacokinetics, which is how the body absorbs, distributes, metabolizes, and eliminates the drug. Most studies enroll healthy volunteers who are screened and dosed under close clinical supervision. Dosing typically begins with a sentinel approach with 1 or 2 people first, followed by the remainder of a small cohort once safety is confirmed. Doses then increase stepwise using a predefined escalation plan and stopping rules. Teams monitor adverse events, vital signs, electrocardiograms and laboratory results, and collect timed blood samples to build exposure profiles. The goal is to identify the maximum tolerated dose, which is the highest dose with acceptable side effects, and a safe exposure range for later studies. These findings set dosing boundaries for subsequent phases and inform early risk management. Phase 1 uses several standard study types, each answering a specific question. Single ascending dose starts with very low doses in small cohorts of often 3 to 6 participants and escalates if safety and blood levels are acceptable. Sentinel dosing and a mandatory observation window of 24 to 48 hours, for example, are common between cohorts. Multiple ascending dose assesses repeated dosing over several days or weeks to check accumulation, steady-state behavior, and longer-term tolerability. Key outputs include accumulation ratios, trough concentrations, and time to steady state. Food effect studies use a crossover schedule, dosed once with a standard high-fat, high-calorie meal, and once fasted, separated by a washout, to see how food changes exposure measures such as peak concentration and area under the curve. Drug-to-drug interaction studies co-administer the drug with known enzyme inhibitors, or inducers, to detect clinically relevant increases or decreases in exposure. Bioavailability and bioequivalence compare formulations or routes. Bioequivalence typically requires the 90% confidence interval for exposure ratios to fall entirely within 80 to 125%. Replicate crossover designs may be used for highly variable drugs. Adaptive escalation methods like BOIN use accumulating data to recommend escalate, hold, or de-escalate decisions, aiming for a target toxicity rate while minimizing time at clearly subtherapeutic or unsafe doses. Traditionally, Phase 1 parts ran sequentially and took time. Integrated or umbrella protocols combine single or multiple ascending dose and food effect work in one adaptive protocol, with predefined decision points. Participants can move between parts, recruitment is streamlined, and data is more consistent. Safety review committees can make go or no-go calls between parts, and operational setup is shared across the whole program, cutting weeks from timelines. Planning a Phase 1 trial is systematic. Start with clear objectives. What safety, pharmacokinetic, and pharmacodynamic answers do you need, and which endpoints will inform dose and label language? Involve statisticians early and simulate dose escalation paths, waiting periods, and sample sizes under different scenarios. Build flexibility with interim reviews and predefined rules to add, pause, or stop parts. Standardize meals, sampling schedules, electrocardiogram monitoring, and data management workflows across sites, and use validated electronic data capture. Define methods, central laboratory handling, and shipping times to protect sample integrity. Document roles for medical oversight and unblinding procedures for emergencies. Even well-planned trials can stumble. Common pitfalls to avoid include relying only on rigid 3+3 escalation, when model-based methods would be more efficient, poor dose selection that is too low, leading to inconclusive data, or too high, risking avoidable toxicity, treating food effect or interaction studies as optional, underestimating washout needs in crossover designs, weak cross-functional communications, and misalignment with the intended product label and patient use. Address these by predefining stopping rules, aligning every study part to a specific labeling or dose selection decision, and scheduling regular safety and data reviews to keep the program on track. Ready to make your next Phase 1 study a success? At Quanticate, we combine smart design, adaptive methods, and operational expertise to get you clear, reliable results. Contact us at inquiries@quanticate.com to discuss how we can help you plan and deliver a Phase 1 trial that's safe, efficient, and built for regulatory success.